Hi everyone, welcome back to part 2 of the cloning workshop. Last time we learned how to actually design and define our experiments and design our primers. It was more of a nuts and bolts episode where we actually sought to understand the methods that we will be using and the operations that we can perform. In episode 2, what I would like to do is how to actually use the primers that we designed for PCR, how to troubleshoot the PCR, as well as how to gel extract our products and how to actually assemble them together into your end product. As well, we will cover the different types of homebrew mixes for TIDA, Gibson, etc., as well as for KLD and for Golden Gate assembly. So hopefully you've been enjoying this series so far, and just let me know in the comments below. Alright, so we've designed our cloning experiment and our primers. Now you're asking yourself the question, how do we actually generate and assemble these fragments? And the steps are more or less the same for KLD mutagenesis and overlap assembly, but we'll go through them step by step. Essentially, we have to first prepare the fragments in terms of generating them through PCR, and then finally we have to assemble the fragments together using a variety of different types of enzyme mixes. So for KLD mutagenesis, essentially we are going to perform PCR on our template with our 2 mutagenesis primers that we designed. We will perform quality control just by checking it on a gel. We may do an optional gel purification depending on your results. And finally, we'll use KLD mix to circularize our mutant vector. For the overlap-based cloning techniques, essentially we just have more PCRs that we need to do. First of all, you likely need to do PCR on your destination vector to linearize it. You need to synthesize your inserts with PCR or get them synthesized by a company. And then you're going to do the same quality control all on a gel. Make your decision on how to purify and go forward, whether it's through gel purification, spin column purification, etc. And then finally setting up your assembly reaction with the various mixes that you can make yourself. As we talked about previously, KLD mutagenesis is essentially a one fragment assembly reaction. So all we need to do is do one PCR, and as long as we get a PCR product of the right length and sequence, then likely our KLD mutagenesis reaction is successful. So this is how I would approach setting up the PCR. If you are experienced, then you can certainly deviate from what I have here, and I, as always, encourage you to experiment with the different types of methods that I mentioned. So for KLD mutagenesis, we are performing a PCR usually of very large plasmids, all in one go. So you're going to want to use the most highly accurate polymerase that you have available to you. Personally, I find Q5 to be the best polymerase out there right now. The bottom line is that you want a polymerase that has a high enough fidelity that it won't introduce mutations in the plasmid that you're circularizing. If you're performing mutagenesis of very long plasmids, for example, if you're a plant biologist and doing PCR on the Picambia backbone, the GC enhancer buffer is very useful for generating the correct PCR product. When you're setting up the PCR reaction, remember to use a low template amount, anywhere between 0.1 and 0.5 nanograms of plasmid per 25 microliter PCR reaction, as this will make it easier in the end for the DPN1 in the KLD mix to actually degrade the template plasmid. Trust me when I say that you don't need more than 0.1 to 0.5 nanograms of plasmid, and you can actually go far lower than that. Combine the buffers, template plasmid, primers, and DNTPs as per the recommendations of the manufacturer of the polymerase that you're using, and then use these PCR settings as a guideline to generate your PCR product. So these cycling parameters are based on Q5, however you can modify them for your polymerase. Usually we do an initial denaturation for 90 seconds, followed by 30 cycles of 95, your annealing temperature, and then 72 for 30 seconds per kb, as that is long enough to generate most PCR products. The annealing temperature will vary greatly depending on the primers that you choose, anywhere from 50 to 67 degrees Celsius. Determining the optimal annealing temperature is critical to generating the proper PCR product with a minimal of background. Traditionally, this is quite laborious to do, as you would need to run multiple PCR reactions one after another. However, if you have a gradient PCR machine, what I would recommend you do is you set up a gradient PCR of 4 to 6 reactions across your heat block, ranging from the most common annealing temperatures of 55 to 67. This ensures that at least one of the annealing temperatures that you choose will be the correct one to give you a good PCR product. If you don't have a gradient PCR machine, then I would start at 59 degrees Celsius and pray to your favorite deity of choice. We'll look at a gel in just one second. However, if at 30 cycles you're getting a weak band, you can bump up the number of cycles to 35 or even 40 cycles. However, keep in mind that the more PCR cycles you do, first of all, you're going to reach a plateau above which you can't exceed any more product. As well, more cycles usually mean more non-specific bands, so it's just something that you need to watch out for. And then finally, after cycling, I like to do a final extension at 72 for twice of the cycling extension time, just to finish off any half-complete plasmids. After that, let your PCR machine just coast to room temperature. I have shown that PCR products can survive for days at room temperature in your PCR machine, so there's no need to use your thermocycler as a refrigerator and wear out the Peltier elements. So in terms of quality control for a KLD Mutagenesis PCR, these are the general steps that I follow. First of all, I take 5 to 8 microliters of my PCR product and I just resolve it on a 1% agarose gel. And then I can ask the questions: do I have the product of the expected size, and is the product relatively clean? Here are 2 real KLD mutagenesis reactions that I've done, and I've highlighted the expected and desired product with a red asterisk. As well, for both reactions, I performed an annealing gradient in the hopes that one of the annealing temperatures would give me my desired product. If your KLD PCR reaction looks like the figure on the right, then congratulations. It's nice and clean, and usually you don't need to do any sort of cleanup in terms of silica column or gel purification before going on to the KLD reaction. So if you have a product that looks as good as this, chances are you're good to go. If, on the other hand, you have bands, non-specific bands, that are as abundant as your desired product, chances are, if you did a KLD reaction based on this product, very likely you would have to screen many, many more colonies. The most conservative and safest choice if your gel looks like this is to optimize your primers, design different primers to get your gel looking like this. You can optimize your PCR in terms of different additives, but essentially the safest bet, if your PCR looks like this, is to optimize either your PCR or your primers so that it looks nice and clean. What you can also do is you can pool multiple samples of your annealing gradient, if you did one. You can gel purify your specific band that you think is your expected product, and then perform your KLD reaction. And generally speaking, if this is your desired product, then this will also work quite well. If you like to live dangerously, you can also perform KLD on the unpurified PCR product and then simply screen more colonies. However, the amount of colonies that you screen to by chance get your expected product could be quite high and couldn't be not worth the time that you think that you save. However, I leave that choice up to you. As we mentioned in part 1, NAB's KLD mix, the Q5 Mutagenesis kit, is quite a nice kit. However, paying $5 to $20 per reaction is quite a bit of an ask. However, this homemade KLD mix that I have here works really great, and I've been using it for years. And thank you to MSR 2009 and Reddit for the recipe that I originally got this from. So to assemble the reaction, you take 1 microliter of your clean PCR product, or 5 to 10 nanograms of a gel purified band, and you combine it with T4 DNA ligase buffer, T4P and K from NAB, T4 DNA ligase from NAB, and DPN1 from NAB, with the remainder just being water. You incubate this whole mix after mixing gently with pipetting for 1 hour at room temperature, and then you just transform 1 microliter of that 10 microliter reaction into 100 microliters of your favorite competent cells. And that's essentially it. If you got a decent PCR product, then KLD reactions are generally quite smooth. So hopefully after transformation, you walk in the next day and you have colonies on your KLD mutagenesis plate. We will get into how to screen your desired product after we get through how to prepare the fragments for Gibson and Golden Gate assembly. Preparing your fragments for Gibson or Golden Gate assembly is similar to KLD in that you want to use a highly accurate DNA polymerase like Q5. As well, you can consider using GC enhancer buffer for longer fragments. However, for shorter fragments, which are less than 2 or 3 kb, keep in mind that you may or may not actually need GC enhancer. Don't just use it by default, because it actually lowers the fidelity of your PCR by just a little bit. You also need to use a low template amount, just like KLD, to make it easier for the DPN1 to digest it. And the PCR cycling conditions are very similar to the KLD mutagenesis, except I would highly recommend that you do a gradient PCR with anywhere between 4 and 12 reactions. And this is because the downstream purification of your fragment will have you losing quite a bit of yield from your PCR product, and at the end of the day, you want a high enough concentration of your fragments to actually do your assembly with. So anywhere between 4 and 12 reactions, pooled together and purified, will give me enough comfortably for my assembly reactions. And while it seems initially more wasteful to do this many PCR reactions from the get-go, keep in mind that repeating your PCR over and over because you didn't have enough yield at the first time is even more wasteful, because now you've spent the additional time and you also feel dumb. So the QC for Gibson and Golden Gate is similar to KLD in that you run a small amount of the PCR product on an agarose gel and then look at the quality of your PCR. If your fragments look something like the figure on the left, where it's primarily your fragment of interest without any other significant bands, then good job, you're ready to go to DPN1 treatment and further purification. The most conservative path that you could take is going through and gel purifying each one of these fragments and then performing Gibson assembly. This is by far the least riskiest protocol. However, it is a little bit slower than the other purification methods. DPN1 treatment of your fragment followed by PCR cleanup with a silica column is a little bit riskier in the sense that you may not get rid of bands that you don't quite see on the gel. However, it is significantly faster and is a decent option if your PCR product looks reasonable on a gel. If you're truly a risk taker, then you can go with the no cleanup option, where you use the PCR product straight from the PCR machine as input for your assembly. This is the riskiest and the most likely to fail. However, it is by far the fastest method of cleanup, in that there is no cleanup. If, on the other hand, your PCR for one of your fragments looks like the figure on the right, which is a real Gibson gel, there's 2 things that you can do. First, you can pool these reactions, DPN1 treat them, and then cut out the band of interest with the gel extraction protocol. Very often, this will give you enough of your expected band that you can comfortably clone with, and is a path that I often take. If you have the time, you can also, just like KLD, you can optimize your primers as well as optimize the PCR to try and get your fragment generation to look something like the figure on the left versus on the right. Since Gibson assembly and other overlap techniques are, generally speaking, more than 2 fragment assemblies, they're a little bit more complex to set up. First of all, after the purification of choice, you want to determine the concentration of all your fragments with the spectrophotometer, whether that be a nanodrop or a cuvette-based one. Then you want to convert the nanograms per microliter reading from your spec to femtomoles per microliter for all of your fragments. And one tool that I found that is particularly good at this is the NAB biocalculator, specifically the dsDNA masked moles tool. When you're performing a 2-fragment assembly, where you just have a vector and an insert, generally speaking, you want to have a 1:3 molar ratio of vector to insert. And if you're performing an assembly of a vector and a small insert, small being anywhere between 100 and 500 base pairs, then you want to actually increase the molar ratio to a 1:5 or even a 1:10 ratio, because the exonuclease in these different types of overlap mixes can actually chew away 1 to 500 base pairs fairly easily. If you're doing an assembly with one vector and 2 or more fragments, then a equimolar 1:1:1 ratio is appropriate. You want to be assembling approximately 200 femtomoles of all fragments combined. I have done assemblies with as little as 50 femtomoles, however that's pretty grim. And if you're doing 3 or more fragment assemblies, I would highly recommend having 400 or even more femtomoles in your assembly reaction. As I mentioned in part 1, there are quite a few overlap enzyme mixes, some of which you can homebrew yourself, and some of which are commercially available. The simplest of these is called the TITA cloning mix. And essentially all TITA cloning mix is some buffer, some DTT, some PEG, and T5 exonuclease, which you purchase from NEB. And you make up this 5x stock and you store it in the minus 80 degrees freezer. And that is essentially all you need to do your assemblies. Generally speaking, TITA performs best with 1 to 3 fragment assemblies. And I found that greater than 3 fragment assemblies has quite a sharp decrease in efficiency. However, the main advantage of TITA cloning mix is that it is extremely inexpensive, costing just cents per reaction. If you want a slightly higher cloning efficiency, or cloning up to 4 fragments, then you can actually make your own 2x Gibson assembly master mix. And it's a bit more complicated of a mix to make, in that you need to make the isothermal start mix first, melt it together, and then combine it with your different enzymes, and then aliquot and store. I'd also like to point out that the homemade 2x Gibson assembly master mix, although far less expensive than the commercial variants, is several times more expensive than TITA cloning mix. So if TITA cloning mix is a few cents per reaction, homemade Gibson assembly mix is maybe $1 or $2 per reaction, and then it goes up from there. It's important to note here that the 2x Gibson assembly master mix, as well as the commercial master mixes, have a notoriously short shelf life. I would say 2 months or 3 months after opening, the efficiency will drop drastically. I would just like to thank Ethan Ford from his Ethanomics blog for the recipe and optimization of his 2x Gibson master mix. You can also buy commercial Gibson master mix to do 1 to 4 fragments, as well as a slightly higher assembly efficiency than TITA. And then truly the creme de la creme of these overlap mixes is the NEB Hi-Fi assembly mix. What makes this mix so special is that they use optimized and mutated enzymes that are an upgrade over those available in Gibson Master Mix. You can look at the patent concerning Hi-Fi Mix for more details, but essentially they do a lot of mutagenesis to squeeze as much performance out of the mix as possible. As well, they include some single-stranded binding proteins, which are not present in traditional Gibson Master Mix. All these assembly reactions have you combine your fragments that you purified in your specified ratio, and then incubating anywhere between 30 and 50 degrees for up to an hour. After that hour is up, you are essentially ready for transformation, and as I said, we'll get to screening in just a little bit. I'm still fairly new at Golden Gate assembly, but here's what I have found that works for me. Essentially, you want to determine the same femtomole per microliter concentration for all your fragments using the NAB biocalculator. Except here for Golden Gate, a 1 to 2 molar ratio for a 2 fragment assembly is often recommended. And then if you're doing 2 or more insert fragments, then a 1:2 to 2, etc. ratio is recommended. I have also seen it reported that equimolar ratios can also be used. Unlike a traditional ligation reaction, since we have a blend of a restriction enzyme and a ligase, both of which work at different temperatures, then we actually need to cycle this reaction to get efficient digestion and ligation over and over and over. So you set up the reaction with your T4DNA ligase buffer, your vector, your insert, your ligase, and your restriction enzyme, and you bring up the total volume with water in a total volume of 20 microliters. And then you perform the following cycling, alternating between 37 and 16 degrees C. It's important to know here that cycling is often recommended strictly for more than 2 or 3 fragment assemblies. However, I found that it doesn't hurt to do this cycling for even just a 2 fragment Golden Gate assembly. It doesn't seem to hurt it. And I've seen cycling programs range from the extremely simple of just 2 simple cycles, 2 different temperatures, to many, many different stages of cycling. So you can make it as complex as you want. Enzymes other than BSA1 are also available for NEB. They seem to specialize in these Golden Gate enzymes, and some of them require slightly higher incubation temperatures. I would just like to go over my gel purification protocol that I've refined over the last few years. And although it may seem like a bit overkill, I have found that by going through gel purification with products that I use for assembly and doing it in this specific way, in the end has saved me more time than I would have gained by skipping it. Essentially, you start off with pooling your PCR products from multiple reactions, and as I mentioned, anywhere between 4 and 12 25 microliter reactions is appropriate. You can certainly try and gel purify one reaction, but I would say good luck actually quantifying that much after you put it through a gel purification. After you pool your PCR products, add between half a microliter and 1 microliter of DPN1 to your pool reaction per 100 microliter of your PCR product, and then just incubate it at 37 degrees for at least an hour, and overnight doesn't seem to hurt it if you want to be extra sure that all the template is dead. Then you want to actually desalt your PCR product with the PCR cleanup protocol that I have available in the resources using the silica column cheat sheet. You can use your generic silica columns and homemade buffers. I learned this the hard way, as you can't just concentrate a PCR product with precipitation or a centrivap and run it on a gel. You'll end up with a smear on an agarose, the low melt agarose, because you have essentially also concentrated the salt in the buffer as well as your DNA. You also want to avoid loading too large volumes on an low melt agarose gel, because you will essentially be diluting your PCR product in the gel volume, and you'll have to cut out a bigger gel slice and process that. So what the PCR cleanup protocol with the silica columns does is it both desalts your PCR product and it concentrates your pooled PCR product. And trust me when I say that the yield from the silica column for desalting and concentrating is far, far better than using G50 resin columns. Just trust me on this. Once you go through the PCR cleanup protocol, you're going to want to elute in a specific volume. If you don't have one of these beautiful centrivaps, then you want to elute in 35 to 50 microliters. However, if you do have a centrivap, then elute in 100 microliters and concentrate down to 35 microliters using this beauty. The way that this machine works is that it draws a vacuum, it centrifuges your liquid, and it heats up the chamber all at the same time, thereby rapidly evaporating any liquid that's inside your tubes. This machine has single-handedly replaced ethanol precipitation for me. I can't remember the last time I legitimately needed to do ethanol precipitation since we got one of these. Once you have your cleaned up PCR product, then you want to cast and load a low melt agarose gel. So if you don't have a gel comb, if you don't have a custom CNC gel comb that will fit 35 to 50 microliters, then simply what you do is you just tape a few lanes together using regular tape, and that will enlarge the lanes enough to load 35 to 50 microliters. I would avoid using amphibian bromide and UV light, as this combination damages DNA, and if you're purifying especially long DNA fragments, that means you're going to get more damage over time when under the UV light. I found that my cloning efficiency increased drastically when I switched from UV and ethidium bromide to blue light reactive dyes like Midori green. After casting your gel with your blue light reactive dye, you want to run your DNA until it's properly resolved, and then cut out your desired band and purify with a silica column, again using our homemade buffers with the silica column cheat sheet. Same as before, elute in 35 to 50 microliters if you don't have a center VAP, and then elute in 100 microliters and concentrate to 35 to 50 if you do. And then essentially you want to determine the concentration of your fragments with a spectrophotometer and decide whether you need to bulk up your experiment or whether you're ready to go forward. If your concentrations of one of your fragments is less than 10 nanograms per microliter, then potentially you can go forward with your cloning reaction. However, it's a fairly dicey proposition. I have done and successfully done these cloning reactions with less than 10 nanograms per microliter. However, it is definitely worth considering at this point, taking a step back and performing additional PCR reactions to bulk up your concentration. When you're in the 10 to 30 nanogram range, generally speaking, this is a good enough yield for an assembly of most sizes of fragments. Anything above 30 to 100 nanograms is comfortable, and if you have over 100 nanograms per microliter, then truly you're laughing, because that is enough for comfortably multiple reactions worth of cloning. Before you go ahead and transform your assemblies, it's important to think about what types of assembly controls you should include. Generally speaking, the more controls that you include during your transformation, the easier it will be for you to troubleshoot when things go wrong. If you're doing a particularly difficult assembly, do more controls than you normally would, and it'll help you in the end. So for KLD mutagenesis, you usually want to include the same reaction with the same PCR product for your real KLD reaction. However, this time exclude the ligase. And what this will essentially test for is the efficiency of the DPN1 in degrading your template. If you get colonies on the control plate without ligase, it means that some of your template plasmid has slipped through the DPN1 treatment, and then you need to adjust for that, either by increasing the amount of DPN1, the incubation time, or checking to see if your enzyme is functional at all. For Gibson and Golden Gate assembly, usually I like to have each fragment by itself combined with the Gibson assembly master mix and processed as the real experimental reaction. So for example, you would have the vector alone, fragment 1 alone, etc. This tests for DPN1 template degradation as before. So if you have a large amount of background on plates which are only transformed with a single fragment assembly, it means that some of your template plasmid is slipping through the DPN1 treatment. If you're sure that your DPN1 treatment was successful, and if you're still getting colonies on your control plates with the single fragments by themselves, what this could mean is you're getting self-annealing of the ends of your fragments to themselves and giving you colonies on your control plate. The way that you would adjust for this is, if possible, you would redesign your primers to another location, to a region which didn't anneal with itself. As well, for every transformation and all assemblies, it's not a bad idea to transform your competent cells with a known good plasmid that you have verified before, so your standard PUC19 or pCambia, etc. And what this will test for is either bad plates, bad antibiotics, or bad competent cells. These simple problems are very easy to fix, either by replacing your antibiotics or making new cells, ordering new plates, what have you. Paradoxically, these simple problems are also the ones that can lead you down months-long rabbit holes trying to troubleshoot all the other parts of your cloning experiment, whereas it was just the wrong antibiotic or the bad antibiotic the whole time. So the take-home is, the more effort that you put in to your cloning controls from the get-go, the easier it will be in the long run when things don't go according to plan. So once you've actually performed your assembly, it's time to transform your cells. The transformation protocol for KLD mutagenesis and Gibson/Golden Gate are almost identical, except that for Gibson assembly or Golden Gate assembly, you may want to transform a few microliters more, whereas 1 microliter of your KLD reaction will often give you exactly what you need. But what you may discover is that trying to transform more than 3 microliters of your Gibson reaction in 100 microliters of competent cells will actually decrease the transformation efficiency, and that's due to the presence of PEG and other inhibitors in the reaction. As well, if you're trying to chase higher transformation efficiencies for, let's say, 5 or even 6 fragment assemblies, you may be trying to electroporate your competent cells. What you'll find out also very quickly is that trying to transform more than 1 or 2 microliters of these complex mixtures will cause arcing, sparking, and explosions. The more salt that you introduce in your electrocompetent cells, the less resistance that you have between the contacts, and the higher the chances that arcing will occur. So just be careful. I hope you found the second part of the workshop informative, and we'll finish up in part 3 where we cover how to actually screen the colonies that we get and verify that we have the plasmids that we were trying to make in the first place. Thank you for watching, and I'll see you next time.